Inoculum propagation. The inoculum propagation for viruses Y and X must take place prior to the extreme resistance tests. It is recommended 40 days before planting the test material. The virus Y is satisfactorily propagated in plants of Nicotiana occidentalis, while the virus X is propagated in plants of Nicotiana glutinosa. A total of 150 plants are sufficient for the inoculation of 80 clones or 60 progenies, each consisting of 100 to 200 individuals. The process of inoculum propagation begins with the preparation of a seedbed of sand and moss with 1-to-1 ratio, in which the seeds of each species are sown. 25 days later, the seedlings are separated and transplanted to small containers using the same soil mixture. They remain here for a period of 10 days, after which a second transplant is made from the containers to 4-inch pots. The plants of both species will be ready for inoculation 5 days after the second transplant. Next, prepare the necessary material for the inoculation. As inoculum, we will use samples of infected leaves from previous propagation. First, take 1 sample of infected leaves with the virus that is to be propagated and macerate it in a mortar, adding cold water, aiming for a weight-volume ratio of 1:20. This may also be done using a small, resistant plastic bag, folding the open end in order to seal in the inoculum. Moments before the inoculation, the plants are sprayed with an abrasive-like silicon carbide and are inoculated by softly spreading the viral solution on the leaves with a cotton swab, being extra careful not to cause any wounds. This procedure is carried out in the same fashion for the propagation of virus X in plants of Nicotiana glutinosa in the same stage of development. The Nicotiana glutinosa plants cannot be used as scions in tests for inoculation through grafting with the virus X. Therefore, an additional propagation of inoculum is carried out with this virus in potato plants of susceptible varieties. In the same manner, 40 days before the planting of the material to be tested, a group of 50 tubers of susceptible variety is planted. 15 days later, the plants are inoculated following the same procedure but using gauze. 20 days after the inoculation, the Nicotiana plants and potato plants will reach the necessary stage of development in order to carry out resistance tests. The Nicotiana occidentalis inoculated with the virus Y will display symptoms of mosaic, rugosity, and curling of the leaf margins, as opposed to a healthy plant. The Nicotiana glutinosa plants inoculated with the virus X will display mosaic and chlorotic spot, as opposed to a healthy plant, while the potato plants of the susceptible variety will mostly display mosaic symptoms.